Hello, everyone. This is Senior Biotech Analyst John Vandermosten. Welcome to our channel that educates the life sciences investor on exciting advancements in drugs, biologics, and devices. For more content and news like this, subscribe to our channel below and like or share with others. Welcome back to our discussion today with Alarity Therapeutics and its management team. We first talked to CEO Jim Cullum, and now we're going to introduce you to one of the co-founders of the company, Dr. Steen Knudsen, who developed the drug response predictor platform. Steen, welcome to the show. How are you doing?
Hi, John. I'm doing fine. Thank you.
Great. It's good to have you here. So the drug response predictor, or DRP platform, it takes a holistic look at a patient's genetic profile to match the right patient with the right drug. How does it work, Steen? And it's a type of precision medicine, but maybe you can expand on and talk about how it's a little bit different and goes the next step in the precision medicine space.
Yes. Most precision medicine approaches so far have been based on looking at a single biomarker, a single gene. Our approach is different. At Alarity, we look at a combination of up to hundreds of genes that differ between tumors that respond to a drug and tumors that don't respond.
Okay. And what are some of the common biomarkers that are used in precision medicine, and then how does it compare to the DRP platform, which you said looks at multiple ones? What are some of the common ones that you'd probably see out there?
Yes, there are some common ones that have been in use for many years. The hormone receptor status biomarkers are used in breast cancer. HER2, KRAS, EGFR, they're all examples of single gene, single biomarker approaches in precision medicine.
And I think Alarity's approach actually uses up to 400 different biomarkers to find a good match. Is that correct? Yeah, I'll just let you take it from there.
Yes, we use up to 400 genes, and the reason that we look at that many genes is that cancer is complex, and the mechanisms of resistance and sensitivity are multifactorial. And this is especially the case for drugs that have multiple targets, such as Alarity's multi-tyrosine kinase inhibitor, Davitinib. So by looking at a large number of genes, Alarity's DRP gets a much more complete picture.
Can you explain the process, how DRP works? I believe you first take a biopsy from a patient and then you examine it. Walk us through that process of how a patient is evaluated using DRP and drugs selected that will be optimal for them.
Yes, it is based on a biopsy of the tumor that is sent to a central lab where the up to 400 genes are measured, and then a prediction is made based on looking at those 400 biomarkers. And I can also mention that we have the 400 genes have been identified by comparing tumor cells that respond to a drug to tumor cells that don't respond to a drug. That's how we have identified all the genes that are relevant to determining whether or not a specific drug works for a specific tumor.
And is that part of the validation process, seeing which genes are positively correlated and negatively correlated to success?
The positive and negative correlation of genes, that's part of our development process, and then we validate it in clinical trials where we use the DRP in clinical trials to select patients that respond and show that they do benefit much more from the drug than patients that are negative for the DRP biomarker. And because the DRP biomarker does not select for adverse effects of the drug, then the benefit-risk ratio is improved. And that is very important to the patient, an improved benefit-risk ratio.
Yeah, it sounds like the real benefit is if there are 100 patients and it only works in about 50 of them, you're not even using it. DRP is actually able to select out those 50 or so that wouldn't work in any way, so they can actually move on to a drug that's better for them in the first place. Is that correct? Is that a correct assessment?
Yes, that's exactly correct.
Okay. And one of the things you brought up that differentiates your type of precision medicine from a single biomarker precision medicine is I think the mechanisms of resistance. DRP actually focuses on that. Is that right? And what are some of the mechanisms of resistance that arise that DRP recognizes?
Yes, some of the typical biomarkers I mentioned before are typically mechanisms of sensitivity, but it's also important to take into account all the mechanisms of resistance. There are many known drug transporter genes that transport the drug out of the tumor cell. There are different drug modification and escape mechanisms. So a single drug can have many resistance mechanisms, and with our holistic approach, we identify all of them.
Great. Yeah. And you said holistic approach there, and I think that's a good way to think about it. Are there some cases where DRP does not work, where it's less effective?
Yes. For example, cancer vaccination therapy and CAR T and such approaches that rely exclusively on using immune cells, they don't lend themselves to the DRP platform.
Okay. And I wanted to wrap up this segment just asking about the regulatory approach. This is a drug-device combination. How does that differ from just having a drug get approved by a regulatory authority?
Yes, the regulatory approach for a drug that has a DRP as a companion diagnostic is as rigorous as it is for other drugs, but it then has the added component that the companion diagnostic is approved separately by the FDA as a medical device by the FDA's device division.
Okay, great. Well, Dr. Knudsen, thank you so much for explaining DRP and how it works. I feel much more enlightened now. Again, we have Alarity Therapeutics Chief Scientific Officer, Dr. Steen Knudsen, and ticker symbol ALLR.